Good morning and welcome to part 2, session 4 of the Society of Australian Genealogists in Search of My Irish Ancestors event. I'm Vanessa Casson, your host for this session, Finding Your Irish Ancestors, My DNA Journey with Veronica Williams. Veronica is a self-described enthusiastic and obsessed amateur genealogist with a special interest in DNA research. Veronica was instrumental in getting the SAG DNA research group established in 2015. She does volunteer work on wikitree.com, is a Wikitree ambassador and project leader for the Australia Project. She is also the project administrator for a number of family tree DNA projects, including the Cassidy and Coat surname projects, Britons of Ireland, and County Wexford. Veronica is joining us from Alice Springs this morning, and I would now like to welcome her to our event and hand over to her to tell us about the breakthroughs she was able to make with an Irish problem using DNA analysis. So good morning, Veronica.
Good morning, Vanessa, and thank you for that lovely introduction. Happy to be here. My story today is about finding my Britton ancestors in Fermanagh, Ireland. I'm going to talk about both traditional and genetic evidence that I've used in terms of finding family. It's not a DNA talk per se, but I will be talking about some DNA concepts and tools just to give people a bit of a flavour of what I needed to do to reach my goals. So if people are listening who haven't got a background in DNA research and are interested in learning more about these tools, I would recommend that you come to our DNA research groups that the Society holds. So today I'm just going to go through, first of all, my traditional research and things that I discovered along the way. Let you know some of the questions that I've identified where only DNA can provide the answer. My goals for this particular family in relation to DNA, some limitations of DNA concepts that you need to be aware of, as I just mentioned, a bit about the discoveries and the tools I used, and what's next for me. So this is a story of discovering my Britton family, and it's intrinsically linked with the Cassidy family, who many have heard me talk about before. There'll be some information in this session today that you may have seen in other presentations I have done. So bear with me, those who have seen some of the Cassidy information. There is some repeat here, but hopefully there's a lot of new stuff as well. So traditional research can often only get you so far in Ireland, and we all know about the absence of records, and most of us seem to hit a brick wall in the 1800s due to that. So DNA is one way that it can help to fill in some of those gaps for you. Now, generally, the research that you need to do in DNA, if the further back in your pedigree your problem person is, or your target ancestor, the more complex the DNA research. And today's session is going to go through a lot of the complex tools that I have used to make some breakthroughs. Now, many of you would have seen this slide before that Christine Woodlands has shown in many of her talks. And I just wanted to point out that my story is really at the pointy end of the stick in terms of, and most Irish research, if you're going back more than great-grandfather, second great-grandfather, grandparent, I should say, level, you're going to be in the area where you need to use complex tools, and the time that it's going to take you to make your discoveries is going to be much longer. And I sort of think about this like in traditional research. DNA is really just another tool that we use to help us with our family history research. And it's a bit like when we're doing traditional research, you know, in most of the cases, the index may be okay to get you where you want to go, but mostly you've really got to check the original. And this is what I think about DNA analysis. So a lot of the clustering tools and things like that that are available at Ancestry are all very well to give you clues about your where to go for your genealogical research. But if you really want to prove things, you really need to go to the chromosome level, which is like checking the original of a birth certificate, in my opinion. So I don't believe you can go back that far without getting into chromosome analysis, which is the more complex end of DNA analysis. So a little bit about my research history. I really started researching my family history seriously in about 2006 when I inherited a box of papers from my Uncle Laurie after he died. I had been dabbling in it over the years. I've still got a family tree I drew in about 3rd or 4th class, and was always interested in the family stories and worked with my aunts and uncles to know about the family. But I really got into it at that time in 2006. I then, not long after that, my husband and I moved to the Territory for a few years, and it was while I was in the Territory, actually in Alice Springs, remarkably, where I am today, was when I completed the SAG research certificate. I did my first test with DNA, family tree DNA. It was the only one available in Australia at that time in 2010. I later tested with Ancestry when they released that product in Australia. It was very difficult to get an Ancestry test before that. I tried through America, but with various contacts, but couldn't get it happening. So I think around about that time, it was around about the time that the research group started as well, I discovered this program called Genomic Pro, and they released a Mac version, which is what I use, and I started working with that program. And the reason I mention that specifically is that I believe, and many people have heard me bleat on about Genomic Pro before, and probably all rolling their eyes, but to me, it was the thing that made me start being able to get some breakthroughs on my Irish, particularly on my Irish line, by using that program. Really, it's an organisational tool. Anyway, I'll talk a little bit more about that later. So in 2006, when I inherited Laurie's box of paper, we knew that Rebekah Cassidy was my great-grandmother, and she was born in Australia in Glen Innes in 1852. And I always thought she was Irish. In fact, I always thought my grandmother was Irish, the way everybody talked about being proud to be Irish. But I discovered that I had 2 generations of Australian born, my grandmother and my great-grandmother, and it wasn't till my second great-grandparents were from Ireland. And there were 4 of them, so on my mother's side, so that was good. But it was a bit of a surprise to me. And Rebekah was the daughter of Thomas Cassidy from Fermanagh and Mary Sweeney from County Clare. But this is the story of the Cassidys and the Brittons. So my Australian family was extensively researched by Uncle Laurie he started in the 1950s. And it was difficult to get back to Ireland in those days, as many of you would know, in terms of having to travel overseas to get information, not as easy as it is today. So Laurie's research mostly focused on descendants of Thomas and Mary Sweeney. And he's had remarkable networking across mostly New South Wales and Queensland, where the descendants were. And I've got a lot of certificates and things for the extended family, which has actually been very useful in my DNA research. But he didn't go back to Ireland. So what we knew in 2006 was that there's me at the bottom of the tree, and going up, my mother, my grandmother, my great-grandmother Rebekah, her father Thomas, and all we knew was that his father, that was named Stephen Cassidy, and his mother was surname Britton, but unknown Christian name. Now, the first thing that Laurie had found, and in this, I believe, came from the Catholic Weekly, was that Rebekah had a cousin who was the late Venerable Archdeacon Cassidy. Now, this is her obituary. And so this was a clue that gave Laurie information about extended family of Thomas Cassidy and Rebekah Cassidy, his grandmother. And Philip Cassidy was actually, his title, Venerable Archdeacon, I understand, is something 2 steps off a saint. But he was posthumously declared heroic in virtue, mainly for his work with the Indigenous population in Maruya. But there's some extensive information, as Jill has already alluded to this morning, about priests, and we found quite a bit of information about the extended family through Philip Cassidy. Now, his father turns out to be Philip Cassidy, Sr., a second son of Stephen and unknown Britton. Now, the death certificate of Thomas shows unknown Britton. We hope that the death certificate of Philip might show something else, but it says Christian name not known. Both have the fathers listed, Stephen Cassidy. So we still don't know the name of our Britton ancestor. Around that time, my Auntie Mary and I got very involved in researching the Cassidys, and Mary turned up the convict indents and found that the 3 brothers were transported in 1830 as convicts. Now, this was a complete surprise to us. We had not known that they were convicts at all. And it turns out that there was Thomas Cassidy and Philip Cassidy, but there was also a 3rd brother, Edward. Edward escaped the colony in 1833, and we still don't know what's happened to Edward. Thomas also left a wife and 2 daughters in Ireland, who I've been unable to locate either. But 3 brothers were transported. Their crime, throwing the landlord's horse from a cliff and killing it. They were actually evicted from their land in 1826, and no doubt that had something to do with it. The father, Stephen Cassidy, was not transported due to his age, and there's extensive material available, most of which I did find on Find My Past, and also was provided with by good folk in Ireland. And so there's a lot of information about Stephen Cassidy and Thomas Cassidy in particular in the newspaper reports, but there's never been any mention of the mother. Hasn't been named at all, so no clues there. Now, in about 2008, I met this lovely 3rd cousin of mine, Marnie, online as we were researching the same people, and she turned up this manuscript in Queensland, and essentially it's the life of Father Philip Cassidy, and it's really an ecclesiastical work. It's many chapters, but the first chapter is very interesting, is that it has some information about the Cassidy family and references back in Ireland. And you can see here that he talks about an Anglican, one of the daughters of an Anglican church clergyman married an outstanding Catholic, young Captain Cassidy, and blessed with a number of children, and talks about 3 of their sons, Thomas, Philip and Edward, leaving Ireland, sailing to the newly discovered colony of Australia. In all these references, they are talked about as farming, good young farmers, not the convicts that they were. Anyway, we won't worry about that too much. Anyway, so this suggests that there was a number of children, and that only 3 of them came to Ireland. But we really don't know about any other, we didn't know about any other siblings. There's a whole lot of stuff on Wikipedia and Family Search Wiki that you can read about Ireland around this time. But I'm not going to go through all of that, but essentially just to say that, you know, there was a lot of tension between landlords and tenants, as most of you would know, and also Catholics and Protestants. So a lot of the Britton is an English name, and it's given the reference to the Anglican church clergyman. It's likely that the Britton family were Protestant and probably came from England or Scotland. And being in a mixed marriage, so Stephen Cassidy, being Catholic, married to the daughter of an Anglican clergyman, generally known as being in a mixed marriage, and was probably likely to have been socially unacceptable and perhaps even resulting from estrangement from her family. Not long after that, I found Ruth. Now, Ruth and I have been co-collaborators on our Britton Family since about 2008. And it was me asking questions on the Fermanagh Gold website, asking about Anglican clergymen, where Ruth approached me, and we struck up a friendship and have been going for over a decade, researching the Brittons from Fermanagh. And when I first started corresponding with Ruth, I felt like I was talking to my auntie Margaret. She was so just everything about her was familiar, and we felt certain we were related. Now, Ruth's family came from a long line of church sidemen. They're a type of church official, also from Bow Fermanagh, Protestants in the Church of Ireland. And that's her, did I say that? It's just her sitting there in front of the Church of Ireland in Bow. Now, her family resided in Tullyholven Townland, and her ancestor, James Britton, managed the Britton Inn at Bow, and that's now known as the Linnet Inn. That's a picture of it there when I visited in 2011. Sadly, the thatched roof no longer exists, so I'm very lucky to have that photo. Now, Ruth believed that all the Brittons in Bow were from the same family, but lacked the records to prove it. And together over the last, 11 or 12 years, we've been scouring for evidence to connect our families. And we've gathered a lot of traditional research on all the Brittons that lived in Fermanagh around that time. So her family came from Tullyholven, as I said, and the Cassidys, before they were evicted from their land, came from a townland called Gawk. And these two townlands are only 2.5 kilometres apart. So it's highly likely that we could be connected. Now, if you have a look at the Griffith's Valuation in around about 1862 was put together for Bow. I looked in Gawk, and of course there were no Cassidys there because they were evicted from their land. But there are some Brittons in the adjoining townland of Tullyholven. And you can see there James Britton and William Britton. And James Britton is Ruth's ancestor, and William is also a sibling of James. So could these Brittons be part of my family? We don't really know exactly where in Gawk the Cassidys live. There's two options based on some anecdotal evidence from the newspapers about where their house was located. And it's in one of these areas. It was either here or down here. There was two plots that went to a landlord called Andrew Clark. Anyway, sometime after that, we discovered this IGI record, and the IGI record showed Catherine Britton marrying Stephen Cassidy. Now, could this be our couple, we wondered? And it was submitted to Family Search, to the index, by this M-Fear. Now, I spent a lot of time trying to identify this M-Fear and connect to them, but it was about around the time when various privacy things were coming in and it was all getting a bit tight there. So I had no luck for quite some time. We looked at the place and it says Enniskillen, Ireland. So at that time we were thinking that it might mean Ennis, County Clare, not Fermanagh. But we weren't sure about that. And the other problem was that it talks about a marriage in 1809. Now, my Thomas was born in around about 1800, so we thought that was a bit late. So, but we were hanging on to this for a long time, trying to work out whether this was the same couple. Now, in about 2009, a fellow by the name of Don told me that he had it was some type of relative. Mary Fear was some type of relative of his, and she had a Bible that had some information in it. And it turned out that she was this M. Fear that was on the IGI record. Now, he was telling me he was going to get a copy of this Bible for quite some time. Eventually, I found some contact details for Mary on the internet. I was constantly scanning around and trying to do forensic research about finding out who this person was. And eventually I got in touch with her. And remarkably, she was very kind to scan what she had, and she sent me copies of the Bible, and it consisted of births, deaths, and marriages, several pages. I've just cut out the snippets here. And you can see here on this actual marriage one, it's a bit hard to read, unfortunately. It says Stephen Cassidy, Catherine Britton, James Cassidy, and Margaret McElroy. And when you look at how this is written, it's Enniskillen, Ireland. Now, Enniskillen is actually the main town near Bow Parish in Fermanagh. And you can see here that someone's tried to make sense of it, and it's really quite clear here that it's Enniskillen. So it was a transcription error, and given that this was the closest town to Bow, we're thinking that this is probably quite right. And you can see here that they're estimating James Cassidy's birth date to be 1810. So the marriage of 1809 seems to have been estimated. So I nagged all these people to do DNA tests. But unfortunately, so far, what do we know about Stephen Cassidy? We know he was a Catholic captain. We think that was a nickname, not an actual title, because it's unlikely that he would have been in the military. He was evicted from his lands in 1826. Now, there's a fictionalised story about the Cassidys in the Lisborough Gazette, 1879, which suggests he was over 70 in 1835 and had a large family, and was not transported due to his age. So we know there's these three sons, but what do we know about his wife? Not a great deal. We know her surname was Britton. She was the daughter of an Anglican clergyman. Her family probably came to Ireland as part of plantation, was in a mixed marriage, and probably lived in Gortgall before 1826 with her Cassidy family. So we think she was probably born somewhere between 1765 and 1788. It's very unclear about her age. So we know we've got the three brothers. So by 2011, this is where we stood. But is this US James Cassidy another brother? Could our unknown Britton's name be actually Catherine or Kitty? We needed more evidence. DNA seems to be the only way to answer this question, but both Mary and Don seemed reluctant to undertake DNA tests. Mary was getting a bit on in years and couldn't quite understand how I could find anything out about a dead person from her. But anyway, that was a funny conversation we had. So DNA testing. When we talk about DNA testing, I always tell everybody to think about what their goal is with DNA testing, because otherwise you can end up just going round and round in circles and not getting very far. Most people are looking to solve a family dilemma or to validate their pedigree, or some just want to know about their ethnicity and nothing else. So it's the beginning of your journey, and it depends on each individual's where that takes you. So in terms of my Britton goals, I wanted to identify the name of my third great-grandmother, probably born around 1768, probably in Ireland. I also wanted to find candidates to do DNA tests in the United States so that I could see whether James was another brother, and then perhaps her name would have been Catherine. I wanted to find other Britton family in Fermanagh, and in time, I'd like to be able to find out the origins of her mother and need to find a female descendant of unknown Catherine Britton and Stephen Cassidy. Might not be that easy. And finally, to find a little bit more about the origins of her father. So I need a male descendant carrying the Britton surname to take a Y-DNA test. So that's what I set out to achieve. Now, I had a look at Mum's ethnicity results. it is a moving feast, and but I think the estimates are getting better and better as they become more familiar with what they're trying to work with, the databases that underlie the ethnicity estimates. So sometimes, you can't, I don't believe you should worry about the very small numbers like this Norway thing. But when you're getting into 9 or 40 or 48, you really need to look at those and make sure that's aligning with what you know about your own ancestry. So what surprises me about this is that Mum's getting more and more Scottish all the time. So, and we don't have any Scots in our family tree at the moment. So could some of this be contributed to by the Britton family? Now, when you're looking at DNA research, you've got to remember that when you go back in time, the further back you go, the less DNA you share with all those ancestors. And this is a picture of a pedigree chart, and you can see here that the closer the relatives, the darker the segments are. So up to your first great grandparents or the second cousin level, you usually 100% chance of matching with someone at that level. But by the time you get out to the second great grandparents, and that's the third cousin level, mostly you'll match with people, and that's why that's a bit sort of lighter there. 98% of the time you will match with a third cousin. But as you go further and further back from that point, it actually starts to dramatically decrease the chances of you matching with somebody. So it's not that easy, and you may test and you may not have any if your ancestors out here in this white bit here, you may not have any DNA from that person. So you've got to remember that your genealogical tree is very different to your genetic tree. Even though these people are still your ancestors, you may not share any genetic DNA with them. And the key thing is to know your tree, of course, and for most people would know out to third cousins, which is about 30. But as the further you go out, there's more and more generations that you need to know, and you really need to have a complete tree for DNA analysis to work. And my goal is to DNA confirm my pedigree out to this fourth great grandparent level, which is the sixth generation and fifth cousins, so that you can build a solid DNA base. And just like traditional research, you need to work back in time. You can see here that as we go further and further back, the number of ancestors that we have starts to increase dramatically. And unless you know all of those, every person in your tree out to the level of your target ancestor, you may be assuming a genetic match with somebody that's not real. Could be coming from someone else. So tree completeness is essential. I like this slide from Debbie Kennett. She talks about the Ancestor Paradox, that at 30 generations, we all have, if you do the math of 2, 4, 8, 16, 32, as each layer goes on in terms of your ancestor pedigree chart, at 30 generations, you should have over 1 billion ancestors. But the world population in 1000 AD, which would be 30 generations ago, is only 295 million. So the message is, the further back you go, the more ancestors you share with others. And just by having a DNA match and sharing a distant ancestor in your tree is not sufficient to confirm a genetic relationship. Don't forget that the Irish married very closely. And often you can find that the segments that you share with some of your Irish ancestors may be looking larger than they should traditionally because of close relationships further back in the pedigree. So just be mindful of that. Now, I just want to tell you a bit about my relationship with Ruth on that score. So if this James and Catherine, if my ancestor, who we're calling Catherine Britton in this chart, was related to Ruth's ancestor, James Britton, if they were siblings, my mother would be a fourth cousin and I would be a maternal fourth cousin once removed. Interesting thing about Ruth is that I'm also a paternal fourth cousin once removed on my father's side. We share these ancestors, Thomas Ellard and Elizabeth Risley from Northamptonshire in England. So we are potentially double fourth cousins once removed. And if you remember back to the porcupine chart I showed, fourth cousins once removed don't necessarily always share DNA. But we're double fourth cousins, and I was hopeful that we would be able to prove some relationship, and at the very least that Ruth would match with my mother. So I nagged her for quite a long time to do a DNA test, which she reluctantly agreed to do in 2014. Unfortunately, she didn't match me. She didn't match my mother or any of her siblings. And by that stage, I think I'd only tested 2 of Mum's siblings. I've now tested 4. She still doesn't match any of those. She didn't match my mother's maternal first cousin, Michael. But she did match a third cousin on my paternal side. And I also share a genetic connection with that person. And I believe we've proven that we're connected on the paternal side genetically through that match. But we didn't actually share any DNA ourselves. But we're not talking about that side, the Ellard side of the family at the moment. Now, sometimes later in that same year, Michael's nephew, Mum's first cousin, so my second cousin, John, tested and they matched. Hallelujah. So if John and Ruth, John has the same sort of relationship with Ruth in terms of the pedigree, that he's related through my mother's sister, Eileen, and Michael, who's Mum's first cousin's sister, Mary. So the relationship is fourth cousins once removed, and they shared about 31 centimorgans. Now I'm going to talk a bit about chromosome analysis now, but I won't be going into it in too much depth. But for those of you unfamiliar with DNA, we all have 23 chromosomes. And as you know, we inherit DNA from both the father and the mother, but we only get 50% of each. And we've got 23 chromosomes, and so there's a process called recombination, and how it's handed down to children and siblings is random and can be different between them. So there's 23 chromosomes is what you've got to be aware of, and that the mix that's handed down each time can be different on each chromosome. Now, women have 2 X's, which is also, and men only have 1, and that's sometimes called chromosome 23. And I will be talking about chromosome 23 or the X chromosome shortly. So this is a snapshot from GEDmatch of the match between John and Ruth. So John's my second cousin and Ruth's my partner in crime. And but having a match, a one-on-one match at that level, fourth cousins once removed, is good. But really, we need more evidence to show that this is the relationship we want it to be is actually the one that it is. So firstly, for John, we need to know whether it's a maternal or paternal segment, and it needs to be maternal for Britton to be possible. And the same and with John, he didn't match any other known relatives in this area. And Ruth, we didn't have anyone to compare Ruth with in terms of any other testers from her family. And it needed to be paternal for Britton to be possible. Now, this next slide, this next little graph here, this blue and pink line here, is an extract from DNA Painter. And I like DNA Painter because it shows you these little lines here, which are indications of a false positive region. Now, false positive regions are where, false positive regions are where it could be a false match. It could be an old population segment, which many people would share, and so I looked at that. It's only at the front end of that chromosome, but it could explain why that chromosome is 31 centimorgans, and really the true match is beyond those little stripy lines. So, and the key thing that we need to do when we're talking about a match between fourth cousins once removed is not to just assume that that's where the relationship is, but getting a third person who also what we call triangulates on that segment that also shares a similar ancestor. So without that three-prong approach to triangulation, you cannot be sure that this is coming from that particular ancestor. We hope so, but we don't know. And some of you have already discussed this particular chromosome back in 2015 in one of our workshops. I used this as one of our examples. So what is triangulation? As I said, it's three cousins all matching each other, and they need to match on the same chromosome in the same segment area from the same ancestor. And it needs to be from different descent lines. So it needs to be coming from sort of like the red area, the green area, and the purple area, for say, so that they're separated, and it's clear that they are all coming from the same ancestor. This is particularly important for conformations greater than third cousins, and particularly in Ireland where no paper trail exists for some of this work. So looking at the match between John and Ruth, I then looked at what is called the matching segment search at GEDmatch. And that gives us lists of everybody who matches in this same segment area. And I scoured that list, and there was probably about 30 names. A lot of them were in this front area, which I'm trying to ignore. And but I really seriously looked at the ones at the back end of the match there to find people's pedigrees and to try and scour those pedigrees to find common names or locations that might be an indication of shared ancestry. Now, a lot of those people didn't reply as usual, which is very frustrating as part of this, but we did get two shared ancestor possibilities that emerged. So there was this Hugh Weir, who was born in 1725 from Enniskillen, and Jane Britton, born 1768, who married Henry Brooks. And these two are John and Ruth, of course. So are these four people related? That would help me prove that this match on chromosome 17 is coming from the same ancestor. You might recall in the 1862 Bow census at Tullyholven, we had the two Brittons living in Tullyholven. But if you have a look closely, there's this Charlotte Weir is also listed, and with William Britton as another landlord in this area. So could this Charlotte Weir have something to do with Hugh Weir? Don't know. Possible, but more evidence is required. Now, a little time later emerges and Bible Don appears in my Ancestry DNA results. I was very surprised to see his name one day. He matched my mother, he didn't match me, and it was only two very small segments. So I was a bit disappointed about that, but I was quickly onto the email and nagging him to upload to GEDmatch. And he did match some of the other siblings, but there were no obvious triangulations. So I was a bit disappointed. But then I forgot, I actually realized I'd forgotten about the X chromosome. At GEDmatch, you do one-on-ones for the autosomal queries, which is chromosomes 1 to 22, but you have to do a separate query for chromosome 23. And remarkably, when I did the queries for chromosome 23, he did match to the other cousins. Now, the X chromosome has a special inheritance path, which is very, very useful in these types of cases. But you can only see the X chromosome at GEDmatch and FTDNA. So if you haven't got your results on those sites, you won't be able to see whether you have any X chromosome matches. Now, men are particularly good because when they have an X match, because they only inherit it from their mother, they do not have an X chromosome. They only have one X chromosome, I should say, because their other pair is the Y chromosome, which makes them male. So this is exciting news for me because this suggests that my Thomas Cassidy and the James Cassidy in the US, which was the source of that marriage record for Stephen and Catherine, are probably brothers. And you can see here, this chart down the bottom is the chromosome map pedigree chart, and the coloured boxes are the ancestors where Don would have inherited X chromosome. And you can see there that Catherine Britton is on that chart. Stephen Cassidy is not. So, and the two Australian cousins, they all match each other as you need to do in a triangulation, and their chart looks exactly the same as this, and they have Catherine Britton coloured in as well. So XDNA has confirmed our Britton's name as probably Catherine. So you can see here a pedigree chart around 2016 where I've now got Catherine Britton, and the three cousins that were involved in the triangulation are shown here. Now the green boxes indicate DNA testers. Now you might think this one over here, the John Roberts and Michael Stevens, is a bit close, but by 2017 I've got 5 people all matching on the X chromosome as well. So that makes me pretty confident that Catherine is the mother of both Thomas and James. So what can we conclude from this point? We're reasonably confident that Catherine, is both the mother of Thomas and James, and therefore my ancestor, the wife of Stephen Cassidy. The X chromosomes matches I just talked about are also supported by other matches on other chromosomes, so that increases my confidence level. Our Cassidy family is likely to consist of 4 sons, but there are probably more siblings. But what about her family? Now, in the extensive written material about the Cassidy family, there is this article in the Enniskillen Chronicle, which talks about Thomas Britton, and Thomas Britton's nephew is Thomas Cassidy. So we know that this is another sibling of Catherine. He lives at Mulligan, is married, has but one son, and his daughter is here. So it seems to suggest that he only had one son and mostly daughters perhaps, or only one daughter, very unclear. But that's all we know. So from Ruth and my research, we have identified 7 or more potential siblings. And you can see there with the asterisks, Thomas and Catherine are the only ones we've got paper records for. Now, all these others are living in the same area. We think that they are actually all related and all siblings. Now, in 2015, I started using Genomic Pro, and it's a database that helps you analyze your segment information, and it's extremely good for managing multiple kits. But it's a complex tool, and it's not an easy learning curve. But I credit Genomic Pro with the success that I've had with my Britton research. So you can see here, this is just one of the examples, and this is a match Karen, who matched on the 23rd chromosome, or the X chromosome. And you can see here, these are all the profiles that I'm managing, different people who are aligned to the Britton family. You can easily see how they all match on this 23rd chromosome and what the relevant segments are. So you can manage lots of different profiles, and it's easy to see how they all link up to each other. And you can document who they inherited the segment from, which can be sometimes tricky on the old brain if you're doing it on a piece of paper. So this is actually Bill, who's one of the people that was in that triangulator group on chromosome 17, and his ancestor is this Jane Britton. Now he also matches Ruth, John and Karen, And you can see that here on chromosome 17. So how does his ancestor fit in? So on chromosome 17, we've got John, we've got Karen, and we've got Ruth all matching. So we believe that Ruth is actually connected to our family from that match. But how does Bill and Jane fit in? We really don't know. Now, by 2019, I'm continually looking at matches of all these people, and I've got probably about 28 different cousins who have all tested, who are contributing to this research. Now, these charts that I'm showing you, the green boxes are the testers. There's not 28 on there. So this chart is actually trying to show now through research how many are actually sharing Britton segments. And you can see here that Ruth's here, and we now have another cousin from her line that also matches in to this family. We have a second cousin, not just Bill, descended from Jane Britton, and there's several others that are descended from Jane Britton that we have sort of isolated matches for, but not triangulations. Now, based on her age, we're not quite sure how she fits into this tree. I've put her in as an aunt, mainly because she's a lot older than some of these others, but she could equally be the eldest sibling. Now, these numbers are meant to represent the chromosomes where they're matching in. But you can see that we've got cousins all over Australia now joining in with this Britton family. Now, I fit over here, but I'm not in this chart yet because I haven't got any Britton segments. This fellow here, John, in 2019 was another sibling that we were getting matches for, but we didn't have triangulation, but we're pretty sure that he was actually going to fit in here nicely as well. One thing that you need to remember when you're doing research like this is it's just like traditional research. You need to start from the current day and work backwards, and make sure that your segment analysis holds up. And that's commonly called walking back the segments. Now, visual phasing can help in that process. I discovered this technique in about 2016. It's really just a logic puzzle, and it helps you to map the chromosomes of siblings back to at least 3 siblings back to their to their 4 grandparents. So we're trying to map my back from my mother and her siblings to her Cassidy grandmother, and Catherine Britton is the maternal grandmother of Rebekah Cassidy. Now, this is just a little chart that shows you what you can do with visual phasing, and these light green ones are all the Cassidy segments that were inherited by my mother and her 3 siblings on chromosome 17. And you can see here that on that top line, they've all got Murphy at this end, which is their grandfather. You can ignore the bottom line because that's the paternal side. So with visual phasing, it's really giving you a clue that you're on the right track if the segments are falling into the same areas that visual phasing is telling you is coming from that particular line. And first cousins can be also useful in this sort of analysis. And our first cousin Michael, what he did or did not inherit can also give you additional information. So you can see here on chromosome 17, I'm mapping Rebekah Cassidy. So I'm doing ancestor DNA painting for each of the 4 grandparents of my mother. And you can see here that at the beginning of the segment, the four siblings inherited segments from their grandmother. But at the back end, there was no segments inherited because they all had the Murphy segment at that back end, as I showed you on the earlier screen. So no Cassidy DNA at the end of chromosome 17 was inherited by the 4 siblings. So John's match is at that back end. The only overlap that there is this little bit here which is in that false positive region that I spoke about before. So I wouldn't worry too much about that. So it fits in nicely there. And when we look at Michael's, so John's uncle, and they both would have inherited from Rebekah's sister Eileen, his are all up the front end of that chromosome as well. So he's got no Cassidy segments down here. So it is likely that John may have inherited a Britton segment at that location. And so this is really a bit of secondary evidence that it's holding up at the Rebekah Cassidy level that it could be a Britton segment. So traditional evidence and genetic evidence go hand in hand. So what you need to do is to keep reviewing the documentary evidence about your family, as well as monitoring the DNA matches of as many descendants as you can find. Since preparing those charts and going through the information for this presentation, I did come across a couple of extra things that I had overlooked before. And the first one was in 1862 in Tullyholven, where William Britton was residing. He was managing a forge. He's listed as, and in the serialized story I mentioned earlier that Peter McGuinness wrote, he suggests that around in about 1826 there were two forges in the town. The busiest one by an Orangeman whose nephew had Papist sympathies. Could this be a reference to our Britton and Cassidy families? If so, it's another little bit of traditional research that gives weight to the DNA confirmation that James and Catherine are potential siblings. So here's how our tree looks like in 2020, and you can see I've got a lot more DNA testers in here. Ruth sits here. I still fit over here. And this is a theoretical tree because clearly we're relying on the segment analysis to show us how these people are connected. And behind all this data, I have got a lot of documentary evidence about each of these chromosomes, but I won't bore you with all that. Just to say that the tree is now growing, but there's no Britton DNA segments identified so far for me or my mother. So the key message is using the DNA results of just yourself may never give you enough information to confirm a distant ancestor. It's necessary to build up the DNA of multiple descendants to be able to work out the DNA profile of your target ancestor or ancestral couple. Just like I was doing with that chromosome map for Rebekah Cassidy, I'm trying to map all her chromosomes so I can use that as further evidence. And consequently, I'm also using all these matches from the DNA analysis I'm doing to map who I'm calling Patriarch Britton and his wife. This is the parents of Catherine Britton. So this couple are the first great-grandparents of Rebekah Cassidy and my fourth great-grandparents. So how I'm mapping this is mapping how the segment was handed down. So what lines of the family received the segment and mapping them here. So any segment, if I've got it right, they shouldn't overlap. They should all just fit in neatly if I've got it right. If I've got it wrong, it'll probably look wrong, or subsequent evidence will emerge that shows that it is wrong by it overlapping somehow. So it needs to consistently build up to this profile. And when you put John's match in there, at the moment, we haven't got any other matches on chromosome 17, but it does fit nicely in there. But we need to be always mindful that other matches or more information that comes to light further down the track, just like with traditional research, needs to be reassessed if it's not looking consistent with what you were expecting. So now, based on all of this, we started out looking for 7 or more potential siblings, and all those people that you can see on this chart with the red dot are who I believe have been connected into this family through the DNA research. Now, we're not quite sure about Jane because, as I said earlier, she could be a sibling or she could be an aunt, but there's clearly a genetic connection with her family. But Marjorie down here, we've got no evidence. We haven't found any. I've got no descendants coming from Marjorie in the research at the moment. So we don't really know if she fits in, but if she fits in and Jane fits in, it seems to be too big a gap. So I would suggest that she would be an aunt, but it depends really on what we find out about Marjorie. We haven't got any testers for my Thomas, even though we know he exists from the traditional research, but perhaps he had daughters who married and changed their names. I finally met Ruth in Enniskillen in Ireland in 2017. We're double cousins who share no DNA. But I think there's a bit of a resemblance there, but maybe I'm just deluding myself. So where to from here? I'm going back to do traditional research. And I think this is the key thing with DNA research, that you move back from traditional to genetic, traditional to genetic all the time. The DNA is giving you more clues. I'm going to be looking at the townland called Lesky, because there's several Britons living in the townland of Lesky. And as you can see, the forge was here, and my Cassidy's lived in Gall, and Lesky is in between those two areas. So they're worth pursuing. So I want to research these three other Britons who were there at the time of Griffith Valuations. And what I would do from that is to look at the valuation notebooks to see how the land was handed down after the Griffiths and into the 1901 and 1911 census, hoping to find some potential DNA candidates in the present day. The other thing you might notice here is that I've noted here some McGuires. And McGuires is a name that's starting to be a big interest to me through other DNA research. And the McGuires and the Cassidys go back as families for generations in Fermanagh. They're very long-standing names. There's lots of them, so it might be nothing. But those of you who heard me talk about the Cassidys back in May, I think it was, I have this emerging group of Cassidy-McGuire DNA matches, and there's a whole lot of them, and some of them are quite large matches. And I was thinking that perhaps this might be my Thomas Cassidy's first wife, who he left in Ireland, but there's a few things that don't add up. Or it could have been Edward Cassidy, the escaped convicts family. Of course, there's Thomas Edward Cassidy here, but I wasn't sure. I still haven't been able to fit them into the family, but it started to occur to me that perhaps these could be connected via the Britons, via the McGuires, and that the Cassidy is a red herring. You might remember in 1828, Thomas Britton, the brother of Catherine, was living in Mullockdun. By the 1864 Griffith Valuation, there's no Britons living in Mullockdun, but there are these McGuires, senior and junior. By 1901 and 1911, there is a Michael Cassidy living in Mullockdun with his family, and he's saying he came from County Cavan, and but County Cavan actually borders Fermanagh, and there's some parts of it that are actually inconsistently named as Cavan or Fermanagh. So could this land have been handed down through the generations, Britton to McGuire to Cassidy? I need to do a lot more research on the valuation notebooks for these particular townlands, and then find a DNA candidate to test. There are also these McGuires in Gall. Even though the Cassidys were ejected from the land, perhaps they are some family connection. So going back to this slide that we showed at the beginning, just wanting to say that I hope I've demonstrated today that the complexities required to go back in time with your Irish ancestors. And researching distant ancestors with DNA, you've got to recognise that it's a long-term and time-consuming endeavour. But there are some tips I can give you. The main thing is to start off at Ancestry, is particularly if your ancestors came to Australia early, because there's so many Australians on the Ancestry database, and you'll find that a lot of connections there. Even though they don't have a chromosome browser, you can still find relevant people to talk to and try and encourage to transfer to GEDmatch or somewhere with a chromosome browser. You can see there that I've got lots of family groups for this research. I've got more people in those groups than any other. And I constantly write to them, updating them on where I'm at, trying to encourage them to go to GEDmatch. But unfortunately, of these 400 odd people, only about 50 are at GEDmatch. So use these, and the other useful place is MyHeritage, and I'm finding MyHeritage is really good because it has the chromosome analysis, but also many people have trees, and I can see MyHeritage growing. They also have a lot of Irish testers there, which can be useful. You can search for Irish testers. So if you want to confirm beyond third cousin matches, you need to undertake chromosome analysis. And this is even more critical when there's no paper trail, such as in Ireland. Things I'd encourage you to do is encourage collaboration, and you can start a project at Family Tree DNA, create a Facebook group to encourage communication about such the family. Blog about it. I've written a couple of blogs about what I've found with the Britton family so far, and hoping to source more people for my project. And create a profile on a One World site like Wikitree and document your research. So I have this profile here, and I've got a whole lot of information about the DNA projects, and in trying to get people to join me. So in terms of my goals, I think I've achieved the first three. Whilst I've connected my Catherine with other Britton family in Ireland, I still want to do more. And I also want to find those descendants so that I can undertake mtDNA and Y-DNA testings, so that I can find a bit more about Catherine's mother and her father. Because if I can get back further on that line, we may eventually discover the name of her parents. So the key messages is finding your Irish family in the absence of records can be done. Chromosome analysis takes effort, but it can be very rewarding. But be prepared to have the dedication for analysis over time. So that's all from me. And as always, this is a long-term endeavour, so I'm sure there'll be more to report down the track. Thank you.
Thank you, Veronica. That was just really amazing to see the level of research that you've gone to. And Carol and Sue and Gail and Christine have all commented that you're very tenacious. And a couple of them have said that they're now inspired to get on with Genomic Pro a little bit more and have another go at it. We just had one quick question, which Brooke has just asked a bit of clarification, that you weren't able to find any baptismal records at all for your Cassidy and Britton families.
No, we have got some baptismal records, but nothing showing the parents of those oldest siblings. So we've got marriages for most of them and then their children, but for Catherine and her siblings, most of those we don't have any baptismal records for them.
Okay.
Does that?
Yep, I think that answers. Brooke was just looking, I think, for a little bit of clarification about your starting points. So.
Oh, my starting points was all the paper stuff for many, many years.
Yeah. So, yep, Helen's also saying it was a very interesting presentation. So overall, I think you've given us all some food for thought, and we're all probably trying to block out big spots in our diary to spend hours sorting out our DNA research. We'll leave it there. If there are any more questions that come through, we'll email them through to Veronica, and Veronica's let us know that she'll be happy to have a look at any questions that do come in through the email. So I'd just like to thank you again, Veronica. It was a very informative session, and it really demonstrated how to successfully combine traditional research techniques with DNA analysis. So thank you for enlightening us all about that. And thank you for joining us from Alice Springs. Hope the rest of your travels are safe. So this brings session 4 to a close. We're now going to have another short break of about 10 minutes. And we look forward to seeing you shortly for part 3, which will be session 5 on your program. It will start at 12 midday in Sydney time. And as with the end of part 1, you will need to actually close out of GoToWebinar and find your link for session 3. So we look forward to seeing everybody again in about 10 minutes for the start of session 3. Goodbye.
Bye.
Bye. Thanks for